Imagine uncovering a hidden diary, but instead of words, its pages are written in tiny fragments of leaves, bones, and charcoal. These minute clues hold stories about ancient lives and environments, often unseen during excavation. Archaeologists process bulk soil samples primarily through a technique called flotation. This specialized method separates delicate organic materials and small artifacts from the soil matrix, allowing us to recover crucial micro-remains that direct excavation often misses. This careful process is vital because these tiny yet invaluable pieces of information are fundamental for reconstructing past environments, understanding ancient diets, and detailing human interactions with their surroundings. They provide direct evidence of what people ate, what plants grew, and which animals inhabited an area, painting a much richer picture of the past. First, soil samples are carefully collected from specific archaeological contexts, often in measured volumes to ensure scientific representativeness. In the laboratory, these samples undergo flotation. The dry soil is gently introduced into a tank of water, sometimes mixed with a chemical dispersant to help break apart clay clumps. Lighter organic materials, known as the light fraction or flot, float to the surface. This material, which includes items like charred seeds, wood charcoal, and tiny insect remains, is then carefully skimmed off using fine mesh sieves, usually with a 1 mm or ½ mm mesh size. Simultaneously, heavier materials such as tiny bone fragments, small artifacts, or uncharred seeds sink to the bottom. This is called the heavy fraction or residue. It is also collected, usually by washing the remaining soil through a series of nested sieves with progressively smaller mesh sizes, often down to a 1/4 mm. Both the light and heavy fractions are then carefully dried, often at low temperatures in a drying oven, to preserve their fragile state and prevent further degradation. Finally, trained specialists, known as paleoethnobotanists and zooarchaeologists, painstakingly sort through these dried fractions under microscopes. They identify and quantify various remains, which can include plant seeds, charcoal fragments, small animal bones, fish scales, and even tiny mollusk shells. This painstaking work contributes significantly to fields like paleoethnobotany, the study of ancient plant use, and zooarchaeology, which examines animal remains from archaeological sites. It is not uncommon for archaeologists to process hundreds of liters of soil from a single archaeological feature to find just a few crucial seeds or bone fragments. These tiny discoveries can reveal astonishing details about ancient economies, climate change, or even specific meal preparations. By meticulously processing what seems like ordinary dirt, archaeologists transform inert soil into a vibrant narrative of human history. This scientific rigor provides an incredibly rich and detailed understanding of ancient life, allowing us to piece together the mosaic of the past, one tiny fragment at a time.